two blocks like core. So the four blocks are block 1 seed and seed storage, block 2 seed viability and seed marketing and treatment, block 3 structure of pollen and ovule seed development, and fourth one seed production certification and seed bank. So if you take block 1, 3 units, seed structure and dormancy, seed storage, and third unit physical and biochemical changes during seed storage. In block 2, fourth unit seed viability and genetic erosion, fifth unit seed marketing, sixth seed treatment. In block 3, seventh one structure of pollen and ovule, and eighth unit principle of hybrid and seed production, ninth unit seed development heterosis. In last block, fourth one, unit 10 seed testing, unit 11 seed certification, unit 12 seed bank. Okay, so this is the total units. Now we will discuss the first unit, seed structure and dormancy. This is very important for short and long question. Here the seed structure for short and seed dormancy and breakage methods, dormancy breakage method. These are the very important for long question, right? Let's see first one. Right. First, let's see the seed structure. Seed structure, if we take the seed, actually seed development from the ovule. Ovule after fertilization, which is converted into seed, developed into seed. So the same, the ovule structure developed into seed structure. Here the ovule integument after fertilization convert into seed coat. For example, let me draw just a rough diagram for our clear understanding. So this is the seed coat. Seed coat, and inside this embryo is there. Embryo. So this is the embryo, and also the surrounded by endosperm. Surrounded by, let me change color, blue color dots. For example, this is the endosperm. This is completely endosperm. Endosperm. Right. So first, let's discuss one by one. First of all, seed coat. Seed coat. In seed coat, the first of all, it is the external part of the seed. As its external part, it gives the protection, and which is made up of two layers, already given clearly two layers. The outer layer called as in seed coat, this is completely together, this both together seed coat. The entire portion, this is called seed coat. In seed coat, two layers, the outer layer, this one, outer layer, thick, this is very thick layer, and this is called testa. This is called testa. This is called testa. And the tegmen, inner layer, thin layer, inner layer, which is called as stigma. It is called as stigma, tegmen. Right? So these two together developed as an envelope to protect the embryo and endosperm. Where are the embryo and endosperm? And here, let me change color. This yellow line embryo. Yellow one embryo. And endosperm. Already told you the blue dots, which is called as endosperm. right? So these both embryo and endosperm located inside the seed, and the seed coat protection to the both. It is protection from the desiccation of water and mechanical injury and environmental effects, so on. And some these this seed coat also useful for dispersal of seed, not only protection, and also it is useful for dispersal of the seed, right? So this is about the seed coat. Now let's discuss about embryo. The another important part in the seed, embryo, and it is a rudimentary plant part, that means younger, to be developed part. It has the axis. This is the axis, embryo axis, and one tip called as plumule. This is plumule, plumule, and which is responsible for the shoot portion. And other one is known as radicle ear, radicle, which is responsible for root production, root production. And plumule ear, from the stem, plumule stem, from the radical roots are developed. So this is about the embryo, and the portion the portion of embryo axis extended above the cartilage is known as epicartil. So this actually these two called as cartilagens. These two cartilagen, cartilagens, cartilagens, right? So above the cartilagens, this portion, above the cartilage, this portion called as epicartil, and below the cartilagen, this cartil hypocartil, right? So this is the embryo structure of the seed, and the storage food material inside the seed. Actually, already told you in endosperm, it is surrounded by the embryo. It is thick and massive structure and made up of elongated cells containing abundant starch and also protein. On the base of presence of absence of endosperm seeds, it is categorized into two groups: albinous endosperm, exalbinous endosperm. Uh, albinous endosperm means which food materials albinous, food material present. If the albino absent, that is called ex-albinominous. And for example, in albuminous seeds, monocot, wheat, rice, and other cereals. Ex-albinous seeds are in monocots, onion. And in dicot, albinous seeds, castor. But ex-albinous seeds, so pea and other pulses. We can observe clearly in castor seed. Do you know castor seed? In castor seed, if we break the seed, the small white color structure appeared. If you press the white color structure, what happened? The oil like liquids comes out. So this is the complete white color structure called as endosperm. White color structure called as endosperm, right? Okay. Now the seed structure completed, and let's discuss one by one. And the seed coat, embryo, endosperm, and in embryo, cartilagens, both cartilagens. Actually, these cartilagens only if we take the dicot, two cartilagens are present. If we take monocots, only one cartilagen present. If we take the gymnosperm seeds, more than two cartilagens are there. So in another important, in monocot, the scutellum is situated in between endosperm and the embryo. So that is only for the, especially in grasses family of monocots, but not all, right? So this is about the some brief introduction, a brief concept of seed structure, and right, this much enough. And also in our book, given the food structure known as seed, and dicots seeds crop family from leguminaceae, which is not important. But here the only in exam point of view, seed structure. In seed structure, you just write about the seed coat and embryo and an embryo structure and endosperm, such enough, right? So here the different type of fruit structures are given: caryopsis, cyzocot, not. These are not known it, right? Now another important one is seed dormancy. Another important one is seed dormancy. What is dormancy? Do you have any idea? Dormancy, dormancy. If you take in our daily life, you must have listened about dormancy. Dormancy in the simple our language, which is called as sleeping or resting. Dormancy period means resting or sleeping period, right? So now let's discuss about seed dormancy and how did it's what are the reasons for seed dormancy, how we break down the seed dormancy. This is the very important and this important question for long one. And okay, right, what happened during dormancy? During dormancy, the normal physical function suspended or slowed down period of time, right? This much of takes place inside the dormancy seed, right? In the let's see, here in our book given, the seed dormancy can be defined as the state or condition in which seeds are prevented from germinating. Already told you in simple language, resting. Resting means no physiological activities, no physiological activities takes place. So such was so that there is no germination even under the favorable condition also. Actually, the seed not to germinate even in general unfavorable condition. But even conditions favorable in that situation also, the seeds are not germinate because of the such type of seeds in dormancy condition. Now let's see the dormancy types: exogenous dormancy, and another one is endogenous dormancy. First, let's discuss exogenous dormancy. In exogenous dormancy, what happened here? Correct? Yes. Sorry for inconvenience. Only host. Sorry for inconvenience. Some of the technical issues. Right. So in seed dormancy, let me change color. Right. So first, let's see the types of seed dormancy. First of all, exogenous exogenous seed dormancy. Later, endogenous seed dormancy. First one, exogenous seed dormancy. In this, the first one physical and second one is mechanical. First, let's see the physical. So in the seed dormancy, then in physical, the first seed coat are impermeable to water. Seeds are low moisture level and legumes are best example. And also embryo is generally inactive. So these are the dormancy. The first dormancy in physical manner, seed coat seed coat as it is a hard one, it is impermeable to water and also immature embryo and seed, right. So the cause of physical dormancy is the structure of outer cell layer. As it already told you, if the seed coat, for example, the seed coat odd, but all conditions, that means the water availability are there and optimum temperature also there, light also there. These are all available. All are favorable to germination. But in this situation also, the embryo does not germinate. The seed does not germinate because of the seed coat. The seed coat very hard. As it very hard, actually the outer side of the water not allow inside the seed, right? Due to the hardening endocarp, impermeable of water, such seed coat developed during the last stage of the seed development. And the second one is mechanical dormancy. So first one in exogenous, A physical dormancy and B mechanical dormancy. If you see the mechanical dormancy, seed coats are too hard to allow embryo expand during germination. And the causes are the structure of the seed coats or remaining fruit occurs in alive. In nature, coats are softened by environment agents such as acids, guts, microorganisms in warm moisture. Actually, this is we will complete in dormancy breakage methods. So anyway, in mechanical dormancy, also the seed coat are too hard to allow the embryo expand. In physical method, seed coat hard to not to allow the water into the seed. In second, in mechanical dormancy, the same seed coat very hard, but what happened during the embryo development, during the germination, embryo develop. Embryo need to be expand. Embryo need to be expand, but due to the hard coat, the embryo not to expand. That's why it goes to the dormancy period. In this situation also, the all are favorable condition. But due to the seed coat, in both in physical and mechanical dormancy, in both situations, due to the seed hardness of the seed coat, embryo not germinate, the seed not germinate and undergoes to the dormancy period. In physical manner, in physical method, actually due to the hard seed coat, the water not to permit into the seed. In mechanical method also, in mechanical method, even the water enter into the seed, but while developing, the not to expand. In both situation, the seed undergoes to the dormancy period. And chemical, the presence of chemical inhibitors in the outer covering of many fruits and seeds. This occurs undergoes to the dormancy period. Dormancy period and examples apples, fruits and seeds and grapes such as and some of the chemicals and also some of the phytohormones also. For example, ABA phytohormone, right? Abscisic acid, which means the chemical. In the presence of abscisic acid inside the embryo, it now undergoes to the germination retarded. Now, not to permit the germination. In addition, oxygen may be restricted to mucilaginous layer, right? So the overcoming methods also given, but we will discuss in breakage methods. So anyway, in chemical method, due to some of the chemicals, best example abscisic acid. Abscisic acid is the best example. That is the chemical method. And also endogenous dormancy. In endogenous dormancy, let's stop it. The second one, endogenous dormancy. Endogenous dormancy. In this first one, morphological dormancy. So embryo is not fully developed. That means inside. So far, exogenous only the seed coat, that is external seed coat external, and also some of the chemical. Some chemical also the applied external. But inside endogenous, what happened? The embryo is not fully developed inside the seed coat. If the embryo not developed fully at the time of ripening, it need additional embryo growth after the seed. The ranunculus family. So this is the endogenous. In endogenous, mainly immature embryo. Due to the immature embryo, of course, when the fruit separated from the plant, that time already the seed matured, embryo also matured. But in some plant, even before the separated the fruit from the plant, the embryo is in immature state. Already fruit separated from the fruit, seed also separated. But when you try to planting on the approximate soil in adequate temperature and water, but not germinate because all are available, and also seed coat also clear. There is no chemical to restriction, but there is no proper embryo, immature embryo. In this situation also, it is goes to under dormancy period. Now the breakage method. How you break these methods? First one, physical dormancy. After that, that means physical methods and some other treatments are given. First of all, here given in physical dormancy. Let's change. This is very important for long question. Very important for long question for dormancy breakage method. So far, we discussed about the types of dormancy. That means the reasons for the dormancy. One of the reason is exogenous dormancy. Second one is endogenous. In exogenous, both the physical manner and mechanical manner only for reason is the strong hard core. Then endogenous only one is the immature embryo, right? In chemical, the external application of the some of the chemical, example ABA. That's it. Now, how can you breaking this dormancy method? Already, if the embryo is under dormancy period, how can you remove it? The first of all, if physical dormancy, already we know the in physical how the physical dormancy occur due to the hard seed coat, right? So if we break this method, we should remove the hard seed coat to make a soft seed coat, right? So hard seed coat to make soft one. Here we follow one of the method that is called as scarification method. One method is scarification method. In scarification method, just wait. Right, in scarification method, in scarification method, the first of all the seed coat, hard seed coat make soft by acid, by mechanical and biological. Another one is biological process. So the in any manner, there is the concept is the hard seed coat becomes soft. That is the concept. So first of all, in acid scarification, these hard coated seeds placed in concentrated sulfuric acid. Due to the when you place the concentrated sulfuric acid, what happened due to the effect of this acid nature, this hard seed coat becomes soft one, right? So the duration of treatment will vary and depends on the type of the nature of the seed coat. If the seed coat normal hard, we can place it in acid just 15 minutes like that. But it is very hard, almost we up to 3 hours. We soaked these seeds, hard seed coat seed placed in the concentrated sulfuric acid up to 3 hours, even up to 3 hours also. So that is the acid scarification. In mechanical, in what happened in mechanical manner, rubbing nature, we take these hard seeded seeds, one of the bowl or one of the container, and these seeds are rubbed on a sandpaper with help of the mechanical scarification by puncturing seed coat. What happened? And simply said, that means if you take the sandpaper or already you have taken the hard seed coated seeds and also you add some of the soil particles. The soil particles act as a sandpaper. When this both together, when you shake well, what happened due to the rubbing nature, automatically the the rapids occur on the seed coat. Due to the rapid nature, the hard seed coat becomes soft. So it is you apply the mechanically. So that's why it is called as mechanical scarification, right? They are the given the example bitter gourd for sand scarification. In this sand scarification, the sand and seed, both sand 2% and seeds, that mean hard seeded seeds 1% ratio, should be followed, rub against hard surface for 5 to 10 minutes. What happened during this period? Automatically, the hard seeds become soft, right? And the next one, a biological scarification. In biological scarification, biological means in living organisms. It is done by digestion system, animal with help of the gut acids. Of course, in first one acid scarification, you apply artificially. You take the concentrated sulfuric acid in a bowl, and all the hard seeded seeds, hard coated seeds placed in that sulfuric acid. But here in nature, what happened in nature? Some of the birds must have eaten some the seeds. After not only seeds, actually fruit. After eaten fruit, what happened? The food got digested, and even some of the seed coat also digest, but not complete. But when it is desiccation, during the desiccation, these seeds are out by done scarification. Already we know in gut, so different type of acid, especially HCl acid is there. Due to the acid treatment, the automatically hard seed coat becomes soft within the gut of the birds or animals, right? So this any manner we should remove the dormancy. In first manner, in acid, after only one reason is the under dormancy period, the hard seed coat. If you utilize the acids, hard seed coats become soft. In mechanical, if you use the sand, if you use the sand due to rubbing nature, it becomes soft. In biologically, if any animal or bird eaten the fruits, automatically the seeds are making soft. Now another one is second one is heart treatment. In heart treatment, it is effective case of leguminous tree, leguminous seeds. In this manner, the seeds should be soaked in the boiled water for 5 minutes. Some crop like Bengal gram and groundnut hot water treatment for more than 1 minute. That is the based on the seed coat, right? This is the hot water. Is the name itself given hot water, right? Now stratification treatment. Scarification clear. Now the stratification. If what happened during scarification, hard seed coat becomes soft. In stratification, when the seed dormancy due to embryo factor, the seed can be subjected to stratification treatment. That means in stratification, it is subjected to embryo development, but not seed coat. In scarification, only for the seed coat, but in stratification, embryo. The embryo, that means it is for only, especially for immature embryo. For immature embryo, we apply the cold stratification and warm stratification. That is the based on the species. If you take the cold stratification, let me just wait. Cold stratification. Don't get confused between the scarification and stratification. Right. Now, stratification, cold stratification, cold stratification, and warm stratification. So already name given itself cold stratification incubate the seeds at low temperature. Low temperature. That mean 0 to 5 degrees centigrade or a moisture substrate for 2 to 3 days. 2 to 3 days. 2 month, several months. That is based on the species of the seeds. It depends the nature of the seed and the kinds of dormancy. Example, cherry and oil palm seeds. So this both cherry and oil palm seeds, when the dormant period, we apply the cold stratification. And another one is next to B, warm stratification. The name itself warm. So in what warm stratification? In warm stratification, some seeds require the temperature 40 degrees to 50 degrees centigrade. 40 to 50 degrees centigrade for few days. Example, paddy, rice, wheat, so on. So these 40 to 50 degrees centigrade apply for 2 months for seed breaking. So these two are the breakage methods by stratification manner. And another one is leaching of metabolic and temperature treatment. In leaching method, the seeds can be soaked in water for 3 days, but once in 12 hours, fresh water should be changed, avoided fermentation of the seed. So this process called leaching. So in this, first of all, three actually the duration three days, but every 12 hours we can change the water. What happen if we if we allow, if we stay the water the same, there is a chance to fermentation takes place. That's why we should remove for every 12 hours and also add new fresh water. Example, coriander, sunflower comes under this treatment. And temperature treatment, it is look like some of the stratification in temperature also, low temperature treatment and high temperature treatment. So this is the seed dormancy and breakage methods. This is very, very important. Seed structure, short answer, and seed dormancy types or seed dormancy causes and seed dormancy breakage methods. This is the essay question, important one. And if you see the second in it, seed storage, seed storage, seed storage. In seed storage, seed gene bank are there. What is the one of the seed storage one? Next, some of the standards given for the seed gene bank. And after the seed storage, next line is seed packing and seed storage type of containers, storage containers. That is also important one. And after filling inside the bags, now how to give the labeling. So this is all comes under the seed storage. Now let's discuss the first seed gene bank. This is very, very important. Seed gene bank. Right. The seed gene banks are three types, such as short term storage, medium term storage, and long term storage. So anyway, seed gene bank means the storage of the seeds. So why we storage of the seed? The seeds genes banks are the main purpose. The minimize the loss of viability and genetic integrity of the seed material. So after harvesting, if not properly storage the seed, what happens? The seed capability, germination capability decrease. That's why for this object minimize this loss of viability and genetic integrity, we should store the properly of seeds. Now the type of seed storage, that is based on the type of seed. One is short term storage, second one is medium term storage, third one long term storage. First name itself given the short term storage. First one, short term storage. Second one is medium term storage. And third one, long term storage. These three. First, short term storage. These are generally maintained at the temperature 15 to 20 degrees. Let's change color. 15 to 20 degrees centigrade. Storage at the we can maintain, we make sure the temperature 15 to 20 degrees centigrade and related humidity 30 to 40 percentage. Right? The breeder's materials and germplasm material in processing for long term can be stored in this short term storage. So this is one type of method. And second one, medium term. In medium term, temperature should be 0 to 10 degree. That means decrease the temperature when compared with short term. And related humidity also decrease 25 to 30 percentage. Right? And long term, in long term temperature, again 10 to 20 degrees centigrade. There is no control of relative humidity. There is no control of relative immunity, and yeah, only the collections are maintained in these conditions and can maintain the seeds anywhere from 15 to 100 years. Here the duration time, the long term means 15 to 100 years. Here the medium term, almost given here the seeds matter for only the up to 10 years, up to 10 years. And in short term, my name is given is only for the years, 1 year, 2 year, that is the short term, right? So these are the long term, short term, and medium. Now, what are the standards for the seed bank? For the proper, that means seed bank means for the any type of seed storage, whether it is short term storage, medium term storage, long term storage, we follow some of the principles. First of all, seed drying, next seed cleaning, storage container. We make sure they are all perfectly applied. If this anyone miss, even if you storage properly in any type of storage, whether it is short term, medium term, long term, the seeds viability decrease. That's why you should follow some of the principles and some of the standards. The standards are seed drying. We should dry the seeds before storage and container. We should dry the seeds properly. The drying seeds at the 10 to 25 degrees centigrade, and we make sure the percentage relatively humidity 10 to 15%. Some of the seed percentage is there. This percentage requirement for the seed germination. Now seed cleaning. While you filling in bag, you should make sure the seeds also should clean. After harvesting from the crop field, we should clean the seed. After seed cleaning, actually we had applied to drying. Next, storage container, and make sure the storage containers also should be clean. The storage containers are different for the three types of storage purpose. Generally, paper bags and plastic bags used for shorter because only years. That's why we can use the paper bag and plastic bags. The plastic container and plastic pouches are used for medium term, and for in medium term, plastic pouches can use. Whereas in long term modules, aluminum container. Long term means almost to 50 years above. That's why we should use aluminum container, and not single layer pouches, multi layer pouches and polythene bags also used. Most probably aluminum bags most preferable, right? So these are the standards for the seed gene bank, right? And next one, how many members?